Here we can look about the bacterial structure. Shape. Along with the other properties, shape is used to identify the bacteria. It is determined by the mechanism of the cell wall assembly. Here the first one, the bacterial shape usually can be determined with the appropriate staining, and using the right microscope we can determine the bacterial shape. And there are of several types. If it is a round one, we can say it is cocker. If it is like rod-like structure, then it is a bacillus. And in case of spiral, they are cocci and the bacilli. They often grow in the doublets. If it's a doublet one, we can call it as diplococci. If it is in the chain model, we can call it as a streptococci. Cocci that grow in the clusters are called staphylococcus. Some bacterial species are the pleomorphic bacteriotes. Antibiotics that affect the cellular biosynthesis, penicillin, may alter the bacteria shape. Now we can say about the nucleus. In bacteria, the nucleus is generally called the nucleoid or the nuclear body. The bacterial nucleus is not surrounded by the nuclear membrane, nor does it contain the mitotic apparatus. Composition. The nucleus consists of polyamine and the magnesium ions bound to the appropriately negatively charged circular supercoiled double-stranded DNA. Small amount of the RNA and the RNA polymerase and other proteins are also present. While speaking about the cytoplasm, the bacterial cytoplasm contains the ribosome and the various types of nutritional storage granules. It contains no organelles. The bacterial ribosome. This contains the proteins and the RNA that differ from those of the eukaryotic counterparts. So, while speaking about the types, bacterial ribosome have the sedimentation coefficient. They have 70 years and composed of 30 years, 50 years subunit. They contain 16 years, 23 years and the 5 years RNA. Ribosomes engaged in the protein biosynthesis or the membrane bound. Many antibiotics they target the ribosome. They inhibit the protein biosynthesis. Some antibiotics selectively target the 70S ribosome. Example erythromycin. They do not target the 80S ribosomes. Cell envelope. The general structure of the cell envelope is composed of the macromolecular layers that surround the bacterium. It includes a cell membrane and the peptidoglycan layer. Except for the mycoplasma, they do not have these layers. An outer membrane layer in the gram-negative bacteria and a capsule, a glycocalyx layer, and the antigens that frequently induce the specific antibody response. These are the major components that present in the cell envelope. Here we can speak about the cell wall. Cell wall, this refers to the portion of the cell envelope that is external to the cytoplasmic membrane and internal to the capsule of the glycocalyx. It confers the osmotic protection and the gram staining characteristics. In gram-positive bacteria, it is composed of peptidoglycan, teichonic acid, and the polysaccharides. In gram-negative bacteria, it is composed of peptidoglycan, lipoprotein, and the outer phospholipid membrane that contains the lipopolysaccharides. Peptidoglycan. This is also called the mucopeptide or a murine. It is unique to the prokaryotes. It is found in all bacterial cell wall except Mycoplasma. Here we can say the structure. This complex polymer consists of the backbone composed of the alternating N-acetylglucosamine in N-acetylmuramic acid and their set of the identical tetrapeptide side chains. The tetrapeptide side chains are attached to the N-acetylmuramic acid and are frequently linked to the adjacent tetrapeptides by identical peptide cross bridges or by the direct peptide bonds. The beta and the 1,4-glycosidic bond between the N-acetylmuramic acid and the N-acetylglucosamine is cleaved by the bacteriolytic enzyme lysosome. They are found in the mucus, saliva and tears. It may contain the diaminophilic acid, an amino acid which is unique to the prokaryotic cell walls. Tecnic acid. The tecnic acid and the uranic acid. They are the water-soluble polymer. They contain a ribitol or the glycerol residue. These are linked by the phosphodiester bonds. They are found in the gram-positive cell wall or the membrane. Tecnic acid. These are found in the cell wall, chemically bonded to the peptidoglycan. The lipotecnic acid is found in the cell membrane. This is chemically bonded to the membrane glycolipid. These are particularly in the mesosome. Their function contains the important bacterial surface, antigenic determinants, and the lipotecnic acids. They help to anchor the wall to the membrane. They may account for 50% of the dry weight of the gram-positive cell wall. Lipoprotein. These are found in the gram-negative bacteria. This cross-links the peptidoglycan and other membrane. A peptide bond links the lipoprotein to the diaminophilic acid residues of the peptidoglycan tetrapeptide side chains. The lipid portion is covalently inserted into the outer membrane. Periplasmic space. These are found in the gram-negative bacteria. It refers to the area between the cell and the membrane, between the outer membrane. They are hydrated peptidoglycan. They are also as well as the hydrolytic, including the alpha lactamase, which are the specific carrier molecules, and the oligosaccharides. These are found in the periplasmic space. In outer membrane, these are found in the gram-negative bacteria. Their structure is a phospholipid bilayer in which the phospholipids of the outer portion are replaced by the lipopolysaccharides. It contains embedded proteins. They include the matrix purine. Some non-pore proteins are also present in. They are transport proteins for the small molecules. Their function is to protect the cell from the harmful enzymes and some antibiotics. They prevent the leakage of the periplasmic proteins. Lipopolysaccharides. These are found in the outer leaflet of the outer membrane of the gram-negative cells. Lipopolysaccharides, they consist of the lipidae, several long-chain fatty acids. They are attached to the phosphorylated glycosamine disaccharides units and the polysaccharide. They are composed of core and the terminal repeating units. It is negatively charged and non-covalently cross-bridged by the divalent cations. They are used for toxicity. They are also called endotoxin. Their toxicity is associated with the lipidae. They contain the major surface antigenic determinants. They include the O antigen found in the polysaccharide component. Protein secretion. Protein secretion system T1 7SS. They play a major role in the bacteria interacting with the environment and helping to determine the pathogenicity, particularly in the gram-negative bacteria. The distribution. The gram-negative bacteria have the 6 classes of system. Gram-positive bacteria have an additional unique class and some of the other 6 classes. Gram-negative bacteria, they contain more than one type of secretion system in the class. Surface protein. The antiphagocytic proteins, they are external to the cell wall of some gram-positive bacteria. They function as adhesives that facilitate the tissue colonization with the several species. Fibronectin binding proteins and the Streptococcus pyogenes. Capsule. This is a well-defined structure of the polysaccharides surrounding the bacterial cell wall, which is external to the cell wall. The one exception to the polysaccharide structure is the poly-D-glutamic acid capsule of the Bacillus anthracis. Their function is to protect the bacteria from phagocytosis and plays a role in the bacterial adherence. Placocalyx. This refers to the loose network of the polysaccharide fibrils that surround some bacterial cell wall. It is sometimes called the slime layer. It is synthesized by the surface enzyme Their function is associated with the adhesive properties of the bacterial cell and contain the prominent antigenic sites. Flagella. Flagella are the protein appendages for the locomotion and contain the prominent antigenic remnants. They consist of the basal body, hook, and the long filament composed of the polymerized protein called the flagellin. Villi. They are the rigid surface appendages that are composed mainly of the protein called pillin. Common palli. They are involved in the bacterial adherence and the gram-positive cell conjugation. 6 villi are involved in the attachment of the donor and the recipient bacteria in the gram-negative cell conjugation. Endospores. They are formed as a cell survival response to the certain adverse nutritional condition such as the depletion of the certain resources. These metabolically inactive bacterial cells are highly resistant to the desiccation, heat, and the various chemicals. They are helpful in identifying some species of the bacteria. Bacillus clostridium. Endospores germinate under the favorable nutritional conditions after an activation of a process that involves the damage to the spore coat. They are not reproductive structures. Mesophilums. They are the aggregates of the bacterial cells that form in the soil and the marine environment and the surface of the medical implant. Devices. They enhance the nutritional uptake and often exclude the antimicrobials.